Good day, everyone. I am Aaron Ezekiel Heramia, and welcome to the seventh lecture of the biology curriculum entitled Animals. In this part, we will focus on tissues, and under this presentation, we will tackle what is a tissue development, and types of animal tissues. So, first, what is a tissue? In a simple meaning, tissue is a group of similar cells performing a specific function. So recall your levels of organization. A tissue are group of cells that are formed together to perform a specific function. And the study of tissues, specifically animal tissues, is called histology. Tissues are complex and unique for animals. So tissues the animal tissues are very specific to animals only and very unique to animals, and they differ from other multicellular organisms such as plants and some fungi by the following characteristics. So as we all know, the cells of animals do not do not have cell wall, unlike plants and fungi that has cellulose and chitin cell walls respectively. Our animal tissues or animal cells doesn't have any cell wall but cell membrane to protect the cell. Their cells can be embedded in an extracellular matrix. So, what is an extracellular matrix? It is a non-living material that serves as a medium for the cells in a tissue. So, we will talk about this later on. So, next, their cells have structures for intercellular communication. So, it is important for cells to communicate with each other to coordinate their movements and functions. That is why there are structures such as gap junctions and tight junctions for intercellular communication for them to communicate with each other. Also, they possess unique tissues that are absent in plants and fungi. So, some of our tissues that we will talk, some of the tissues that we will talk about later on are very unique to animals only. For example, tissues for movement or muscle tissues are very unique to animals only. Also, tissues for coordination, such as the nervous tissue, is very unique to animals. For other organisms, doesn't have muscles or nervous tissues. So how do animal tissues develop? Practically all tissues originate from group of cells in an embryo called germ layer. So after rounds of many mitosis happening in the reproductive system of a female organism, usually the cells will undergo differentiation, which is a basic life process or basic life characteristic. All cells will differ or differentiate according to the purpose that they will become in the future. That is why all tissues will originate from a single embryo or single zygote, which the tissues will be originated from a group of cells called germ layer. So the process in which germ layers form is called gastrulation. It is the development of blastula, a hollow cluster of cells into primary germ cell layers. So what are the different types of germ layers that we have? We have endoderm, mesoderm, and ectoderm. So these three primary germ layers also varies depending on the organism that we have. So animals can be classified based on the germ layers they form or the number of germ layers they form, either diploblastic or triploblastic. So when an organism is diploblastic, it only forms ectoderm and endoderm, while triploblastic forms three germ layers: endoderm, mesoderm, and ectoderm. So to expound more of the gastrulation, it is the second phase of development. It is the migration of blastocyst cells inward, leading to multiple distinct layers of tissues called germ layers. So the blastocyst cells migrate inward, leading to multiple distinct layers to form. And these are the steps, so you can read them, but we will not gonna talk about it further. So how do what kind of tissues does ectoderm, endoderm, and mesoderm form in the future, or in the future development of a blastula? So ectoderm becomes outer layer of the skin, hair, lining of the nose, mouth, and the nervous system. Endoderm becomes digestive tract, respiratory tract, liver, and pancreas. Now the mesoderm becomes muscles and skeleton. These germ layers becomes our animal tissues that will form later on in the process of development. So now we will talk about the types of animal tissues. So now we will have four basic animal tissues, which are the epithelial tissue, muscle tissue, nervous tissue, and connective tissue. So first we will talk about epithelial tissue. Epithelial tissue is the most abundant tissue in many animals. It makes up most of the internal organs, covers structures, and also lines cavities. Epithelial tissue can be classified based on their cell shape, number of layer, direction, position, and function. So this classification will determine the name and also the function of the epithelial tissue. So please remember that structure depends on the function. So the structure will depend on the function it will facilitate in the body. And it is a rule of thumb in adaptation and biology that structure will depend on what type of function it will carry on in any process in the body. So for example, our nose has two nostrils, has two holes for it to uh, uh, what do you call this? Our nose has two nostrils in order to accept air or allow air to enter our respiratory tract. So just remember that structure it depends on function. So now we will focus on the classification of epithelial tissue based on number of layers. So if if an epithelial tissue has only one layer, it is called simple. When it has more than one layer, it is stratified. Meanwhile, when nuclei are arranged in a different manner to look like it is stacked or stratified, it is called pseudo-stratified. Now, the cell shape. When the epithelial cells are flat, irregular, circle shape, it is called squamous epithelial tissue. When the cells are tube-like, or it looks like dice or something, it has central nucleus, the epithelial tissue will be called cuboidal. When it is tall, slender, and narrow, it is called columnar. When a stratified epithelial cells that may change in shape, for example, in epithelial tissue of the bladder, in which it changes shape when it is filled with urine. The epithelial tissue is called transitional. So transitional is very unique in our bladder, in our urinary system, because bladder holds our urine and it needs to, you know, expand and deflate when it is filled and excretes urine. So yes, that kind of epithelial tissue inside that bladder is called transitional because it changes in shape. Yes. Okay. Next is for direction and location. So we have two. When the epithelium faces the external environment, or for example, the air or the lumen or the opening or the cavity of an organ, it is called an apical epithelium. So under apical epithelium, we have two, the mesothelium and the endothelium. So mesothelium is originated from the mesoderm, and it is the epithelium that covers most of our internal organs in the pleura, which is in our which is the cavity of our lungs and the heart. Lungs only. I'm sorry. Pleura is for the lungs and the respiratory tract. Pericardium, which is the heart, and the peritoneum, which is our gastrointestinal organs. So, mesothelium covers these internal organs, these visceral organs that we have inside of our major body cavities. Now, endothelium is another type of apical epithelium, which it covers, or it is found in our blood vessels, in the arteries, capillaries, and veins. So next, basolateral epithelium, it is exposed to the internal environment, which is the extracellular fluid. So it faces the internal environment, it exposed in the extracellular fluid that we have. So we have many kinds of extracellular fluid. For example, the yes, extracellular fluid. Okay, so the extracellular fluid is the body fluid that is not contained in the cell, so outside of the cells. So it is found in the blood, in the lymph, in body cavities lined with serous membrane, in the cavities and channels of the brain and spinal cord, and in muscular and other body tissues. So we can find this extracellular fluid all over our body, and it is called sometimes the internal environment of our body. So yes. Next is for the function. It is classified based on the function, lining and gland epithelium. So when we say lining, it lines the organ. So the function of the epithelium is to line the organs, to cover the organs only. And next, for the gland epithelium, its function is to secrete and secrete nutrients, hormones, and absorb nutrients that coming from the blood. So we can see here some tissue sample that are viewed under the microscope. So you can see here that the first picture, it is a simple squamous. In here we have a simple cuboidal. This is a simple columnar. This is a simple pseudostratified ciliated columnar. And this is a stratified cuboidal cell. I, I'm sorry, no. This is a transitional. The last photo is a transitional epithelium. So, as I've mentioned, structure depends on the function. Some of structure will always depend on what type of function it will carry on. So, squamous cells are flat because squamous lines, squamous epithelium lines many organs in the cavity. So we can find some squamous cells on our cheek, and they are flat because they line the buccal cavity or the oral cavity. uh, columnar columnar epithelium are tall and slender because they absorb nutrients. And for ciliated epithelium, they have cilia because they trap foreign particles that may enter in a tract. For example, a respiratory tract, it has ciliated epithelium to trap some pathogens that may enter along with the air that we respire or we inspire. Next is connective tissue. It consists of cells and non-living material, which is the ground substance, making up a matrix. So cells and non-living material called the ground substance makes up a matrix, or yeah, the matrix. Its functions are to sup- to it functions as structural support, connection, and protection. It has the following components. So the connective tissues usually has these three components. So cells, fibers, and ground substance. So cells are the main component of a connective tissue because it will not be called a tissue without the presence of cells. So cells are acts as a defense mechanism for the tissue and produces supportive structures such as the fibers. What so now next we will move on to fibers. So what are fibers? Fibers are produced by proteins to give structural support. So the proteins that are mentioned in these fibers are the collagen, uh, what else? Collagen and yes. So collagen, fibronectin, and elastin are some of the proteinaceous fibers that forms in the extracellular matrix. And next, the ground substance. It functions as a medium for process such as diffusion and cell nourishment. So ground substance usually is a non-living material. So it is not a non-living material, which is sometimes an inorganic compound. For example, water in the plasma, it is a ground substance of the blood. So it can be classified according to the following classification. Abundance of fibers, it we have two classification: loose connective tissue and dense or fibrous connective tissue. So loose connective tissue, also known as areolar connective tissue, contains less fibers but abundant in cells and ground substance. Now dense or fibrous connective tissues contains more fibers and less cells and ground substance. So we will have we will just have to remember that when it is loose, less fibers, more cells. When it is dense, more fibers, less cells. And some connective tissues cannot be boxed or cannot be categorized based on their fibers, so they are specialized. So we have four: adipose tissue, cartilage, bone, and blood. Adipose tissue is a loose connective tissue with high adipocyte content. Cartilage much firmer than fibrosity, no blood supply. Bone, also called as osseous tissue, it is mineralized and rigid. And blood, the only liquid connective tissue for protection and transport. So we'll talk about the specialized connective tissue in the next slides. So as we can see here, connective tissue usually uh, functions together in harmony, and they can also be found elsewhere or anywhere in the body. So we can see here the bone, the blood, the cartilage, fibrous connective tissue, loose connective tissue, and the adipose tissue working together to form this kneecap or the knee joint. The knee joint of a mammal. So the bone or the bone tissue forms the bone. The adipose tissue forms the fat padding in the leg of the mammal. Blood is circulated along in this area. Cartilage uh, covers the bone, serves as a covering and protection of the bone. Also, to prevents friction when it is moving, the mammal is moving. Fibrous connective tissue connects muscles together to the bone. Loose connective tissues are, yeah, also uh, connects muscles together. And these are the connective tissues that we have, and they work together to effortlessly carry out their function. And to ensure that the mammal will move and will be secured when moving. And yeah, so these are the difference between loose connective tissue and dense connective tissue. So yeah, so loose connective tissue can be divided into three, which is areolar connective tissue, reticular connective tissue, and adipose. So adipose sometimes can be classified under LCT, but because of its fat content, we can classify also in the specialized connective tissue. So areolar connective tissue has fibroblasts with some macrophages and other white blood cells. It has collagen elastic fibers. It has a jelly-like abundant ground substance. And under it is found under epithelia of skin, mucous membranes, capillaries, and organs. Now, the reticular connective tissues has fibroblasts with many blood cells. It has reticular protein fibers. Reticular protein fibers are made up of type 3 collagen. So, we're not gonna talk about in detail the reticular fibers, but it is made up of reticulin and composed of type 3 collagen secreted by reticular cells. So it has also jelly-like ground substance which is abundant, and it is found on spleen, lymph nodes, and bone marrow. Now, the dense connective tissue can be divided into three types: regular dense, irregular dense, and elastic dense. So, what is the main difference of regular and irregular? So, regular has thick collagen in parallel. So, it is formed in parallel structure, or it appears in parallel. So, when we say parallel, it is like straight lines that are not meeting with each other. It looks like a stripe. So that is the regular dense connective tissue. Meanwhile, irregular dense has no consistent arrangement. So it may vary, the arrangement of the protein fibers or the collagen fibers may vary. So there are no consistency with their arrangement. So yeah. So we will just remember that this connective tissue has less cells. less ground substance, but many protein fibers, making it much denser than the loose connective tissue. So the elastic dense connective tissue, the difference between the regular and irregular, it has mostly elastic fibers with some collagen, making it more flexible and more elastic than the two. So the regular dense connective tissue can be found in tendons, ligaments, and aponeurosis. Irregular dense connective tissue can be found in capsules around joints and organs and dermis of the skin. Elastic dense connective tissue can be found in the walls of large arteries, walls of bronchial tubes, and vertebral ligaments. Now let's move on to the specialized tissues. I mean, the specialized connective tissues that forms are are classified alongside with the dense and loose connective tissue. So they are specialized because they cannot be classified based on the number of fibers they have, and some of them don't have uh, proteinaceous fibers such as collagen and reticulin. So we have four: bone tissues, cartilage, adipose tissue, and blood. So bone tissue is also called as osseous tissue. It's made up of cells called osteoblasts, osteocytes, and osteoclasts. So osteoblasts are the precursor cells of osteocytes, meaning before an osteocyte or mature bone cell will form, it is an osteoblast first. So osteocyte is a mature bone cell. It makes up most of the bone tissue. And osteoclasts are the cells that ingest or im- uh, engulf osteocytes that serves as a repair mechanism of the bone. So it can be classified as spongy or compact. So spongy bone tissue are less rigid than the compact bone. So the compact bone covers or surrounds the spongy tissue. So the spongy tissue contains more blood supply, or it has the blood supply needed by the bone, and the compact tissue have less blood supply, or it contains the right amount of blood supply needed. So spongy tissue is characterized with the trabeculae or the honeycomb-like structure, and the compact bone is mineralized, meaning it has phosphorus and calcium, which makes it rigid in structure. So the site of nourishment for bone tissues are called Haversian canals, and Haversian canals with the osteocytes and these bone cells makes up and makes up a Haversian system. So yeah, that is the bone tissue. Next, cartilage is a connective tissue less firmer than the bone but firmer than the FCT or the fibrous connective tissue. Why what it makes it firmer than the fibrous connective tissue? It has uh, more fibers than the fibrous connective tissue. It is made up of cells called chondrocytes, and these chondrocytes are placed in an area in the cartilage called lacunae. So it has no blood supply. So the distinct characteristic of cartilage it has no blood supply, but it depends on diffusion for its nourishment. So it can be hyaline, elastic, and fibrocartilage. So hyaline from the Greek word hyalos, meaning clear or glassy. It is the most abundant type of cartilage in the body, and it can be found in our bones as our protective structure or protective cushion, cushion for the bone to prevent friction when moving. And it also found in our rib cage. Yes. Elastic is another type of cartilage in which it is flexible, promotes flexibility. So elastic cartilage can be found on our pinna of the ear or our earlobes, which is our external ear, I mean. External and internal auditory tubes, epiglottis, and larynx. So the elastic cartilage is flexible, promotes flexibility, and that's why it has more elastic fibers. And fibrocartilage can be found in our backbone. Next, adipose tissue is a type of loose connective tissue made up of cells called adipocytes. So these adipocytes collect and store fat in form of triglycerides for energy metabolism. So this fat is used for our nourishment, for cell nourishment, and for our energy metabolism. And this, when adipocytes form together to make adipose tissue, it forms what we call our subcutaneous fat in the skin and visceral fat in our visceral parts or our viscera. So this visceral part and the visceral fat and subcutaneous fat forms a protective cushion and a padding which keeps us warm and also absorb shock when we sometimes when we bump a object or when we are in an accident like that. So it absorbs the shock that we may uh, receive from yeah, an accident or something. So it forms as a protective cushion for shock and also an insulation when it is cold. Next and lastly, for our connective tissue is the blood. It is a liquid connective tissue. It is transported by blood vessels. So it is the only liquid connective tissue that we have, liquid connective tissue, and it is transported via our blood vessels. So blood cells can be uh, classified based on the granules or the conspicuous granules that it has, granulocytes and agranulocytes. So granulocytes are erythrocytes, leukocytes, and thrombocytes. Meanwhile, agranulocytes are lymphocytes and monocytes. So erythrocytes are red blood cells. It contains the pigment hemoglobin, making it appear to be red, and it carries oxygen. It has uh, no nucleus and has central pallor, making it to absorb more oxygen, and it carries this oxygen throughout our body. Leukocytes are white blood cells, and its function is to defend our body against foreign materials and pathogens. And thrombocytes are platelets. These are cell fragments that aids in blood clotting. And when we are wounded, it also forms our scabs. So agranulocytes are lymphocytes and monocytes. So lymphocytes are immune cells, which are T and B lymphocytes. So their function is to defend also our body against foreign material and pathogens. Monocytes also, they are they are assigned to protect our body against pathogens. So when monocytes travel outside the blood, or when they go outside the blood premises, it is called a macrophage. This macrophage engulf bacterial cell and other kinds of pathogen, which is a defense mechanism of our body. So next is a muscle tissue. Muscle tissue facilitates movement of the animal by contraction of individual muscle cells. So animals are only taxonomic classification to possess muscle. So only animals contains muscle for as a structure of movement facilitated by the construction of individual muscle cells. So note, some animals do not have muscles. So some, like for example, si anemon, no, Nidaria, Nidaria, the class Nidaria doesn't have any muscle, and they depend on other structure of movement in order for them to carry out locomotion. So muscle tissue can be divided into three categories. I'm sorry, the Nidarians. Nidaria is a phylum, not a class. Sorry. So okay. So muscle tissue can be divided in three categories: skeletal muscle, smooth muscle, and cardiac muscle. So skeletal muscles are attached to the bones. Smooth muscles are found in internal organs, ducts, vessels, and skin. Cardiac muscle are mostly found in the heart. So what are the difference of these three categories of muscle tissue? So skeletal muscles appears to be long cylindrical fiber. They are striated and many peripherally located nuclei. So what causes the striation of skeletal muscle? It is the alternating arrangement of the proteins actin and myosin. So these actin and myosin fibers are alternately placed to form these kind of striation. So myosin are thick, meanwhile actin are thin, making it to appear like it is striped. So the nuclei of the skeletal muscles are found in the periphery or in the sides of the muscle tissue. It is long cylindrical fiber, meaning it is long and is in cylindrical in shape. So the function of skeletal muscle is to facilitate voluntary movement. Voluntary produces heat and protects organs. So it is attached to bones, as we have mentioned a while ago, and around entrance points to the body. Example, mouth and anus. So we also have skeletal muscles in our mouth and anus. For example, orbicularis oris, muscle that surrounds the lips, is a form of skeletal muscle. Cardiac tissue or cardiac muscle is a short branch striated single central nucleus. So it is also striated because of the tight junctions that it possesses, which are called intercalated discs. Their single nucleus are found in the center of the muscle cells in the cardiac tissue. The function of the cardiac muscle tissue is to contract to pump blood. So it contracts in order to pump blood throughout our heart and throughout our body. So is located, which is it is located in our heart. So the smooth muscle tissue are short spindle shaped, no evident striation, single nucleus in each fiber. So smooth muscles are short, They are tapered, meaning they look like tapered. Uh, it is, yeah, tapered. So when we say tapered, wait, let's define what is tapered. So tapered is diminish or reduce in thickness toward one end. So it is thin in the end, then it thickens in the middle, then it becomes thin again in the other end. So yeah, that is the definition of tapered. So no evidence striation because the it has no actin and myosin fibers, and also it has single nucleus in each fiber. So the function of smooth muscle is to carry out involuntary movement. That is why it is found in our internal organs, ducts, vessels, and skin. It moves food, involuntary control of respiration, moves secretions, regulates flow of blood in arteries by contractions. So these are mostly involuntary processes of our body, and smooth muscles are the one who are who i- smooth muscles are the one who are assigned with these processes. So located in the walls of major organs and passageways. So these are the muscle cells or muscle tissues. So we can see here, skeletal muscles are long cylindrical with striations caused by the muscle fibers, actin and myosin. Cardiac muscles also appears to be striated because of the intercalated discs, which are tight junctions for cellular communication and also to for also contraction. And also it appears to be branch. It is branching. Cardiac muscles are uh, contains a distinct characteristic of branching. And smooth muscles are tapered. So it is thick, thin, then becomes thickened in the middle and then thin again. So it has no distinct muscle striations and the nucleus are found in the center. So these are smooth. These are the muscle tissues that we have. Lastly, nervous tissue, it functions in the integration of stimulus and control of response to that stimulus. So nervous tissues are made up of two distinct cell types, the neuron and the neuroglia. So the neuron is the main cell of the nervous tissue. It is assigned in transmitting electrochemical signals. So, for example, when we have a stimulus, the function of the neuron is to transfer this signal all over the body in order for the body to understand or to be aware, become aware of the stimulus that we have. So the neuron has basic parts, the dendrite, cell body, and axon terminal. So the dendrite receives the signal from other neurons. So the dendrite receives these informations incoming from other neurons, and then the electrical electrochemical signal will then travel into the cell body. So the cell body contains the nucleus and other organelles. And then the signal will then travel to the axon. And the axon usually is covered by a an insulation called the myelin sheath. After that, the electrochemical signal will then be transferred in the axon terminal, which delivers the information to the other dendrites of the neuron. Next, neuroglia supports and nourishes the neuron, and they do not transmit signals. So there are many kinds of neuroglia. So we will uh, talk about this in the next slide. So we have six kinds of neuroglia. So four in the central nervous system and four in the peripheral nervous system. So in central nervous system, we have four: astrocytes, ependymal cells, oligodendrocytes, and microglia. And in the peripheral nervous system, we have two: satellite cells and Schwann cells. So astrocytes are the neuroglia forming the blood-brain barrier, which is the blood-brain barrier is the barrier of the brain from the blood supply. So this prevents the entry of unnecessary materials like pathogens and other materials that are not necessary for the brain. So it also provides external support. It regulates movement of ion, nutrient, and dissolved gas in the brain. Also absorb and recycle neurotransmitters. Ependymal cells line ventricles. So they form as the epithelial cell of the brain. They line ventricles of the brain and the central cord or the spinal cord. It also produces the cerebrospinal fluid or the fluid, extracellular fluid in the nervous system. Oligodendrocytes are the myelination or CNS. It provides insulation of the CNS nerves and also provides structural framework. Microglia remove cell debris. So it engulfs pathogens and also other foreign materials. So its function is to remove unwanted materials that may enter or that may invade the nervous system, central nervous system specifically. And in peripheral nervous system, we have two: satellite cells and Schwann cells. So satellite cells surround neurons and provides nourishment for the peripheral neurons. So next, Schwann cells surround the PNS or surround the neurons in the PNS. So it is the most abundant cell in the peripheral nervous system. It is responsible for the myelination or insulation of the peripheral axons. So it participates in the repair process when these peripheral neurons are in injury. So these are the following tissues that we have, the basic tissue types that we have for animals. So that is all for this lesson. See you in the next part of this lecture. I am Iran Ezekiel Haramia and thank you very much for listening.